Welcome student friends in Alka's Botany channel. Today we are going to learn apoptosis, the important phenomenon which is going on in the cell. I am Dr. Alka Inamdr in Alka's Botany channel. Please like, share and subscribe the channel. During development of multicellular eukaryotic organisms and prokaryotic cells growing in culture medium, some cells are to be eliminated deliberately by a natural process. This process of elimination provides essential control over cell number and maintains natural physiological balance in all organs or cells. Such cells are generally unwanted and they are eliminated during embryonic growth, organ formation and tissue development. Such deliberation elimination is carried out by definite genetic program and executed through ordered physiological reactions. Such deliberation and elimination of cells is called programmed cell death, PCD, or apoptosis. Removal of damaged or unnecessary cells, unwanted cells from the system by genetically controlled reaction is called apoptosis. It differs from pathological cell death, which is due to some infection or disease and injury to the cell. In the mechanism of apoptosis, the apoptosis is executed through physiological reactions responsible for definite morphological and biochemical changes. During this programmed death, the nucleus and cytoplasm condense, the membranes disintegrate and produce vesicles, and fragmentation of DNA occurs. The dying cell breaks into number of membrane-bound apoptotic fragments which are rapidly engulfed by the neighboring cells. This is the programmed cell death continuation program in which normal cell shows cell shrinkage where chromatin condensation takes place. After that, membrane blebbing takes place, and due to that nuclear collapse, continued blebbing. After that, apoptotic body formation takes place, which goes to the lysis of apoptotic bodies or cell death. The apoptosis involves the activation of pathway that leads to the death of a cell. This pathway is an active process and depends on RNA and protein synthesis by the dying cell. The activation of programmed pathway can be triggered by different stimuli under different conditions. The common reactions to trigger the pathway activation are removal of growth promoting factor and binding of specific proteins to the receptor substance on the surface of cell membrane. When the protein binds to a receptor, the interaction between the protein and receptor triggers the activation of a specific protease enzyme. The activated protease releases cytochrome C from mitochondria, which cuts off the supply of energy. Such release of cytochrome C and decrease in energy supply together cause the activation of series of proteases whose actions are responsible for destruction of cell membranes, organelles, and cell structure. Ultimately, the destructed cell becomes fragmented into membrane-bound fragments which are engulfed by surrounding cells. This is a removal of growth promoting factors and binding of specific proteins to the receptor substances on the surface of cell membrane, and the activation of specific proteases enzyme. It releases cytochrome C from mitochondria and decreases in energy supply. Together causes the activation of series of proteases, and ultimately the destructed cell becomes fragmented into membrane-bound fragments and it causes cell death. This is the significance of apoptosis It is also observed in some plant organs during senescence and during growth of embryo. Similarly, bacteria growing in culture medium exhibit apoptosis in some cells. This indicates that apoptosis is of universal occurrence in living system and it is not restricted to the animal systems only. The programmed cell death or apoptosis is used to kill the cells for three different reasons. First one reason, for proper development of a system and to maintain internal stability required to compensate the changes in surrounding environment. Second, for defense or as a protective mechanism against stress in culture medium. And third one is for aging or senescence process. The cells which are generally killed by apoptosis are cells which are non-functional or not more necessary for any physiological function. Another one, the cells that are produced in excess than the requirement of an organ. Cells which are not healthy and show improper development, and the cells which are harmful and needs to be eliminated from the system. All those cells are only goes natural apoptosis, which is an important physiological process and biochemical process of a cell. Thank you very much.